Hi everyone, in this video we're going to learn about special senses. So this is the learning outcome, so you can read it at your own time. So in the last video I have talked about the general senses, and in this video I'm going to discuss about the special senses. So we have 5 special senses, and this include olfaction, which is the sense of smell. Second one is the gustation, which refer to the sense of taste. The third one is the vision. Number 4 is equilibriums or balance. And the last one is the sense of hearings. The olfactory organ, which is responsible for the sense of smell, are made up of 2 layers. Number 1 is olfactory epithelium. Second one is the lamina propria. The olfactory epithelium contains olfactory receptor cells, supporting cells, regenerative basal cell, and olfactory glands. So olfactory receptor cells is a highly modified neurons with chemical receptor, known as odorant binding proteins on the cilia. So the odorants actually is the chemicals in the air that bind to that proteins. So now let's take a look on the olfactory pathway. So let's imagine that you are smelling a rose. So the odor of the particles in the air will stimulate the olfactory receptor cells. So the axon leaving the olfactory epithelium collects into 20 or more bundles that penetrates the cerebriform plate of the ethmoid bone. So the first synapse occur in the olfactory bulb, which is located just superior of the cerebriform plate. So the axon leaving the olfactory bulb and then travel along the olfactory tracts to reach the olfactory cortex, the hypothalamus, and the portions of the limbic system. So a certain smell can trigger profound emotional and behavior response, as well as the memories due to the parallel distributions of olfactory information to the limbic system and hypothalamus. So the perfume industry, for example, understand that the practical implications of this connection and works to develop odors that trigger sexual responses. Now, let's take a look on this animation video regarding the process of smelling.
The process of olfaction, or smelling, begins with hair-like cilia that line the nasal cavity. This lining is called the olfactory epithelium. As air enters the nasal cavity, some chemicals in the air bind to and activate nervous system receptors on the cilia. This stimulus sends a signal to the first-order neurons connected to the epithelial cells. The signal is carried by these neurons from the nasal cavity through openings in the ethmoid bone and then to the olfactory bulbs of the brain. The signals then move from the olfactory bulbs along the olfactory tracts to the olfactory area of the cerebral cortex.
So the human olfactory system can differentiate between 2,000 to 4,000 chemical stimuli. But these discriminations depends on the nature of the odorants. So we detect many odorants in amazingly small concentrations. The olfactory receptor can be replaced. This is because of the basal cell in the epitheliums divide and differentiate to produce new receptor cells. So the replacements of new receptors declines with age. As a result, elderly people have difficulty detecting odors in a low concentration. This is because those receptors that remain become less sensitive. So this is one of the reasons why the other people have difficulty in detecting odors in a very small concentration. So in one situation, your grandma might have a tendency to use a lot of perfumes. This This is because she needs to use more in order to smell it. So now let's move on into the another type of senses, which is gustation. Okay, refers to the sense of taste. So gustations provide information about the foods and the liquid we take every day. So the taste receptor or gustatory receptor are distributed over the superior surface of the tongue and the adjacent portions of the pharynx and the larynx. So the most important taste receptors are located on the tongue. By the time we reach adulthood, the taste receptors on the pharynx, larynx, and also in epiglottis have decreased in number. Taste receptor and specialized epithelial cells form sensory structure known as taste bud. An adult has about 10,000 taste buds. The superior surface of the tongue has numerous variously shaped epithelial projections. This is known as the lingual papillae. And there are four types of papillae. First is the filiform, second fungiform, third is the folate, and number four is velate. So this picture showing you the four different type of lingual papillae. Okay, the first one is the velate papillae. Okay, the velate papillae has as many as 100 taste buds. Okay, the second one is the folate papillae. Okay, you can find these folate papillae on the lateral margins of the posterior regions of the tongue. Okay, the third one is the fungiform papillae, so contain about 5 taste buds. The last one is the filiform papillae, which is important to provide frictions that helps the tongue to move objects. There is some evidence that sensitivity to the four primary sensations, which are sweet, salty, sour, and bitter, are actually varies along the axis of the tongue. However, there are no differences in the structures of the taste bud, and the taste buds in all portions of the tongue will provide all primary taste sensation. There are also other two sensations. Okay, one is the umami. So umami is actually a pleasant and savory taste that is a characteristic of a beef or meaty broth or chicken broth. Okay, this taste is actually sensitive to amino acid, small peptides, and nucleotides. Okay, and these receptors actually are present in the taste bud of the velate papillae. So the second one, which is the water receptor, okay, are present a lot in the human pharynx. Okay, and the sensory output of the water receptor is processed in the hypothalamus, and this actually affects the water balance and the regulations of the blood volume. Okay, for example, drinking will stimulate water receptor, which in turn will stimulate the hypothalamus to secrete antidiuretic hormone, which ultimately prevent over ingestions of water. Taste receptor respond more readily to unpleasant stimuli compared to a pleasant stimuli. So for example, we are 100,000 times more sensitive to bitter substance and almost 1,000 times more sensitive to acid or a sour taste when compared to sweet or salty chemicals. So why is this happen? It's because of this sensitivity has a survival value. So the alkaloid food tends to taste bitter, and this includes many poison. Okay, when we ingest poison, we tend to spit them out from our mouth. So in this way, it can protect us from ingesting a harmful substances. So this is one way of example of a protective mechanism. So each taste bud contains about 40 to 100 gustatory receptor cells and many small stem cells. So each gustatory receptor cells extend slender microvilli, which is sometimes known as taste hair, into the surrounding fluid through the taste pore. So now let's discuss about the gustatory pathway. So when you eat or drink something, okay, the taste receptor will respond to that stimulation because the dissolved chemical is in contact with the microvilli. Then the impulse will travel from the gustatory cells through the cranial nerves 7, 9, and 10. Okay, so the sensory afferents carried by these three cranial nerves was synapsed in the solitary nucleus of the medulla oblongata. Then was synapsed again in the medial lemnicus, followed by the synapse in the thalamus. Finally, the information is projected into the gustatory cortex. We begin life with more than 10,000 taste buds, but the numbers began to decline dramatically by the age of 50. So our tasting abilities actually change with age. So this is why many elder people find their food taste bland and not really good. Okay, while when you compare to the children, sometimes they will find the same food that we eat is too spicy. So the threshold for receptor stimulation varies for each of the primary tastes. As I mentioned before, our taste receptor are more easy to respond or quick to respond to the unpleasant stimuli when compared to the pleasant stimuli, and this is due to the survival value. Okay, so as I mentioned before as well, the toxic compounds are often bitter, and ingesting acid actually can damage the mucous membrane of the mouth. Now let's continue with another special senses, which is the hearing and equilibrium. So hearing enables us to detect and to interpret sound wave. Equilibrium sensations inform us about the positions of the head in the space by monitoring gravity, linear accelerations, and rotations. The sensory receptor for the sensations of equilibriums and hearing are protected from the external environment. So these sensory receptor cells are located within the internal ear, where the sensory information is integrated and organized before it is forwarded to the central nervous system. So the sensory receptor cells of the internal ear are called hair cells because it resembles cilia. So we have stereocilia. Okay, this is actually along a microvilli, and another one we have the kinocilium. Okay, this is a single large cilium. The hair cells are mechanoreceptor, which sensitive to contact or movements. So when the external force pushing on the hair cells, their movement distort the plasma membrane, which results in the releasing of the neurotransmitter. In this way, the hair cells provide the information about the directions and the strengths of the mechanical stimuli. So the ear is subdivided into three regions, and these include the external ear, the middle ear, and also the inner ear. So the external ear is the visible portions of the ear which collects and direct the sound wave towards the middle ear. So it consists of auricle and also external acoustic meatus, that also known as the auditory canal. So the skin that line the external acoustic meatus secrete a material known as earwax. So this earwax is very important to keep out the foreign object or small insects. So the earwax also helps to slow the growth of microorganisms and reduce the chances of infection. The canal is actually lined with many small projecting hairs that is important to trap debris. The middle ear or tympanic cavity is an air-filled chamber that separates the external acoustic meatus with the tympanic membrane. It is connected to the pharynx of the auditory tubes and contains three auditory ossicles. The tympanic membrane, also known as eardrums, lies at the end of the external acoustic meatus. The tympanic membrane is a thin, it's like a semi-transparent sheet that separates the middle ear with the external ear. The auditory ossicle, on the other hand, is three tiny bones within the middle ear. These bones connect the tympanic membrane with one of the receptor complexes of the internal ear. The auditory ossicles have three small tiny bones, which are the malleus, the incus, and also the stapes. So the malleus attach at the 3 points to the interior surface of the tympanic membranes. So in the middle ear, there's two small muscles that protect the tympanic membrane and the ossicle from the violent movements under a very noisy condition. The first muscle is what we call a tensor tympani. Alright, so tensor tympani actually important, to protect the ear from a very strong vibrations or movements. So when the tympani, the tensor tympani contracts, it will pull the malleus, medially, and then stiffen the tympanic membrane. So the increase in the stiffness of this tympanic membrane will help to reduce the vibration. Alright, and then the second one is the stapedius muscle, which actually originate from the posterior wall of the middle ear and starting of the insert of the stapes. The contractions of the stapedius muscle, which is right here, reduce the movements of the stapes at the oval window. Now let's move on into the internal ear. So the internal ear contains the sensory organ, which is very important for hearings and equilibriums. And it actually receives the amplified the sound wave from the middle ear. So the internal ear is a winding passageway called a labyrinth. So the body of the labyrinth, protects the membranous labyrinth and its receptor. So the bony labyrinth is a shell of a dense bones that surrounds and protects the membranous labyrinth. This is the membranous labyrinth. Between the bony and the membranous labyrinth flow the perilymph. So the perilymph here, if you can see it here, Alright, is actually a liquid that closely resembles the cerebrospinal fluid. So the labyrinth consists of three parts. Okay, we have semicircular canals, we have vestibules, and also cochlea. So the membranous labyrinth is a very delicate interconnected network of fluid-filled tubes. So these receptors are important for equilibriums and hearings, and you can find this within these tubes. And the membranous labyrinths contain a fluid known as endolimbs here, like this blue color here. So vestibular complex consists of three parts, and these include the semicircular duct, which is within the semicircular canals, and then we also have utricle and also saccule. Okay, let's see the first one. Okay, the semicircular duct uh, has a receptor. Okay, and this receptor inside the semicircular duct are stimulated by the rotations of the head. So next is utricle and saccule, which provide the sensations of gravity and linear accelerations. Okay, the third one is the cochlear duct that lies within the cochlea. Okay, the cochlear duct sits sandwiched between a pair of parallel chambers, and the entire complex make a turn around. Okay, look like a snail. Okay, and their receptor is actually stimulated by sound. So the membranous labyrinth has four parts. So the semicircular duct, utricle, and saccule uh, are part of the vestibular complex, which monitor equilibriums. And yet, on the other hand, the cochlear duct is responsible for hearing. The region in the wall of the ampulla contains or hosts the receptor. Okay, and this place is known as the ampullary crest. So each of these ampullary crests is bound to an ampullary cupula. So this is actually a flexible, elastic, and gelatinous structure. So the at the ampullary crest, we have a hair cell that actually embedded within the ampullary cupula. So when your head rotates in the planes of semicircular duct, the movements of the endolimbs along the length of the duct will push the ampullary cupula to the side, which distorting the receptor process. Movement of the fluid in one direction will stimulate the hair cell, and the movements in the opposite directions will inhibit them. So when the endolimb stops moving, the elastic nature of the ampullary cupula make it returns to its normal position. So even in the most complex movements can be analyzed in terms of motion in three rotational planes. So each semicircular duct responds to one of these rotational movement. So for example, when you shake your head saying no, and this stimulates the hair cells of the lateral semicircular duct. By nodding yes, this excites the anterior duct and tilting your head from side to side, which activate the receptors in the posterior duct. So the utricle and saccule, okay, provide the equilibrium of a sensation, whether the body is in a stop positions or in a moving positions. So a slender passageway that is continuous with the narrow endoliphatic ducts connects the two chambers. So the endolymphatic duct ends in a closed cavity that is known as the endolymphatic duct. So both utricle and saccule, okay, contain hair cells that cluster in the maculae that sense the horizontal or vertical movements. So the hair cell process are embedded in the gelatinous otolytic membrane, in which the surface contains densely packed calcium carbonate crystals, and this crystal is known as otoliths. So when your head is still, the pull of the gravity on the otoliths shifts them to the side, therefore disturbing the or distorting the hair cell process and stimulate the molecular receptor. So the changes in the receptor activity will tell the central nervous system that your head is no longer level. So this mechanism also accounts for your perceptions of a linear acceleration as when your car speed up suddenly. Okay, another example, when the otolith is lag behind and the effects on the hair cell is can be comparable to tilting your head back. The cochlear duct or the scala media, okay, lies between the pair of the perilymphatic chambers, okay, which is the vestibuli and the tympanic duct. So both ducts connect at the tip of the cochlear spiral, creating one long chamber which begins at the oval window and ends at the round window. So the hair cells of the spiral organs are arranged in the series of longitudinal rows. They lack kinocilia, and their stereocilia are in contact with the overlaying tectorial membrane. So when a portion of the basilar membranes bounces up and down in response to pressure wave within the paralimbs, so the stereocilia of the hair cells are pressed against the tectorial membranes and distorted. If the amount of the movements increases, more hair cells and more rows of hair cells become stimulated. So this pressure wave within the paralimbs are triggered by sound wave that is originally arrived at the tympanic membrane. So the sensory neurons will relay signal through the spiral ganglions to cochlear branch of the vestibulocochlear nerve 8. So hearing is the perceptions of sound. But what is sound? Okay, it actually consists of wave of pressure conducted through a medium such as air or water. So in air, each pressure wave consists of a region where the air molecules are crowded together and adjacent zone where they are farther apart. So the frequency is the numbers of waves that pass a fixed reference point, such as the tympanic membrane, in a given time. So the frequency of a sound is measured in terms of the numbers of cycles per second and has a unit called hertz. So wavelength and frequency are inversely related. Okay, what we perceive as the pitch of a sound is actually our sensory response to its frequency. So for example, a high pitch sounds, okay, it actually has high frequency and short wavelength, might have a frequency of 15,000 hertz or more. When we compare to a very low pitch sound which has low frequency and long wavelength, could have a frequency of 100 hertz or less. Intensity determines how loud the sound is. So the greater the energy content, the larger the amplitude and the louder the sounds. So the sound's energy is reported in decibel. So the flexibility of a basilar membrane varies along its length. So pressure wave of a different frequency affects different parts of the membrane. For example, the tip near basilar membranes responds to high frequencies, giving a high-pitched sound. So let's watch this video to understand the flexibility of basilar membrane.
The membrane's stiffness decreases from base to apex, the base being about 100 times stiffer. When a sound wave pushes the footplate of the stapes at the oval window, endolymph is displaced within the scala media, the chamber containing the basilar membrane. Movement of the endolymph makes the basilar membrane bend near its base, starting a wave that propagates toward the apex. The distance the wave travels depends on its frequency. This high-frequency wave vibrates the stiff base of the membrane a good deal, but most of the energy is dissipated before the wave propagates very far. Now notice the difference in the traveling wave generated by a low-frequency sound. This generates a wave that travels all the way up to the floppy apex of the membrane before the energy is dissipated. These vibrations in the different parts of the basilar membrane establish a place code for sound frequency. This is because different locations on the membrane are maximally deformed at different frequencies.
So let's watch a video showing the process of hearings.
Hearing occurs when sound waves are changed to neural signals inside the cochlea of the inner ear. When the eardrum vibrates from sound waves, a chain of 3 tiny middle ear bones moves: the malleus, incus, and stapes, or hammer, anvil, and stirrup, together called the auditory ossicles. The arrangement of these bones forms a lever system that amplifies the energy of the sound waves before it reaches the cochlea. The stapes fits into the membrane-covered oval window at the base of the cochlea. Within the bony cochlea is the cochlear duct. Alongside the cochlear duct are the vestibular duct and the tympanic duct. All 3 ducts are filled with fluid: endolymph in the cochlear duct and perilymph in both the vestibular duct and tympanic duct. When the stapes pushes against the oval window, pressure waves in the perilymph travel up the spiral through the vestibular duct and back down in the tympanic duct. Pressure is relieved when the wave reaches the round window. When looking at a cross-section of the cochlear duct, specialized hair cells are shown being struck by pressure waves in the perilymph. The endolymph-filled cochlear duct now appears as the central triangular section. The vestibular duct and tympanic ducts, both containing perilymph, are on either side of it. Sitting on the basilar membrane of the cochlear duct are hair cells of the organ of Corti. Above the hair cells is the tectoral membrane. When the basilar membrane is moved by pressure waves in the perilymph, hair cells are pressed against the tectoral membrane, and their stereocilia are distorted. The distortion generates a signal that travels through the cochlear branch of the vestibular cochlear nerve to the brain, where the signal is ultimately interpreted as a sound.
And let's take a look on the neural pathway in the video.
How do neural signals travel from hair cells in the organ of Corti to the primary auditory cortex? Well, let's examine the ascending auditory pathway. Why ascending, you ask? Well, ascending means information goes to the brain, while descending means information goes from the brain elsewhere. Inner hair cells in the organ of Corti are connected to type 1 spiral ganglion neurons, whose axons represent 95% of the cochlear nerve. So firstly, signals travel along the cochlear nerve to the cochlear nuclei in the brainstem on the ipsilateral side. Ipsilateral means same side, and contralateral means opposite side. Here is where it starts to get a little complicated. Most auditory information crosses over. However, each cerebral hemisphere processes stimuli from both the ipsi and contralateral sides. This is advantageous for 2 reasons. First, if you get brain damage in one hemisphere of the brain, your sense of hearing doesn't suffer much. Second, as humans, we process some pretty complicated sounds, like those involved in speech, and getting input from both ears to both hemispheres allows for more processing potential. Anyway, back to the ascending auditory pathway. Our information is at its first stop, the cochlear nucleus. From here, most of the neurons cross over to the contralateral side. This is the primary pathway that the information takes. However, there is also a secondary pathway, in which some neurons stay on the ipsilateral side. In both cases, the neurons synapse in the superior olivary complex, which is also in the brainstem. The signal continues to be relayed along the lateral lemniscus to the inferior colliculus in the midbrain. From the inferior colliculus, the information is relayed to the medial geniculate nucleus of the thalamus. Most of the neurons taking this trip stay on the ipsilateral side. However, some cross over. Finally, the information continues into the auditory cortex. The auditory cortex is tucked into the lateral sulcus. The auditory core region contains the primary auditory cortex, or A1, which is organized tonotopically. In other words, it's arranged by frequency.
Now let's discuss about hearing disorder. So hearing disorders can be either conductive or sensory neural. So sensory neural hearing loss is actually result from damage to the cochlea or nerve pathway from the internal ear to the brain. This can be caused by exposure to a loud noise, head trauma, and aging. So with age, the tympanic membrane become less flexible, the articulation between the auditory ossicle was stiffened, and the round window may begin to harden. So motion sickness often results from a mismatch of a visual and equilibrium cues. For example, while riding in a car, visual input indicates that we are moving forwards, but we are actually stationary inside the vehicle. So the equilibrium input does not perceive as much as the movement. So when you have a motion sickness, the symptoms usually include headache, sweating, flushing of the face, nausea, and vomiting.